So I would like to just kind of kick this off, and so a comment made in the first presentation on the behavioral health endpoint struck me as of interest in terms of caffeine being something that people, in some instances, people actually seek out, and that what is an adverse effect in the situation of exposure to caffeine and behavior. Some behaviors, I would imagine, would be considered adverse, but others, things like alertness and awakeness, may be of benefit in certain situations. And I wonder if we could just have some discussion of what it means to be adverse for a compound like caffeine in terms of dietary exposure.
Yeah, I definitely like to comment on that. The caffeine is consumed ad libitum by populations throughout the world, not just in the United States. And when individuals consume caffeine, they are aware of the effects. They apparently choose to consume caffeine because of the behavioral consequences. Although the research on that is not as quite clear as we might like, but we know a lot about patterns of caffeine, and we know, for example, that 70% of caffeine is consumed in the morning when individuals are trying to wake up, and we know that after that, consumption of caffeine decreases dramatically. So we actually have population data which tells us something about the fact that caffeine is used for particular purposes, and we also have population data which shows that the doses the average individual consumes are inherently limited. So over the last, going back perhaps 40 years, we know that caffeine intake levels have been remarkably stable, which again is indirect evidence that it's the behavior that is why individuals are consuming caffeine and that they're self-limiting their intake. How you match that up with concerns about safety is a very difficult question, and there needs to be, I think, more discussion about how to weigh risks and benefits of something that the population chooses to consume in generally safe levels with concerns about adverse effects.
Thank you for that. I have another question. It's reassuring that the NARAT earlier conclusions are being supported by this systematic review, and FDA, of course, has a long history with caffeine, and in fact, our founder, Harvey Wiley, was involved with looking at caffeine in the early days. And I'm thinking, okay, now we've got, looking at these health outcomes, there are some results that show that there are certain, with these particular endpoints, there are challenges in terms of the exposure data and how granular the data are and how they're collected, perhaps how timely they are. And I'm wondering whether there are some recommendations that could come out of this systematic review that could perhaps guide future research so that some of the studies and conclusions that we have here, we don't find that the research community is repeating studies and repeating the same studies, and perhaps we could help guide the future research on caffeine forward. If there are some highlights that maybe each of you could identify from your areas of expertise in the review, I think that would be beneficial. So.
So one of the areas that was identified was how much variability there is in tolerance between individuals. And so as we think about going forward and we're talking about personalized medicine and knowing what's right for me, I think that the more research can identify how to identify those specific subpopulations that maybe do have to worry about levels that are lower than this, as well as those who can freely consume at levels above what the recommendation. So this whole idea of one value for all healthy adults is an interesting challenge as we learn about inter-individual variability.
Well, as long as I have a chance, I'll just say that I studied tolerance to caffeine quite a lot, and most of it, well, consistently it's acquired. So I think the rate at which people acquire tolerance to caffeine is probably genetically determined. But as I said when I was discussing the reinforcement studies, it's amazing how sensitive some people are to the reinforcement value of caffeine, whereas other people are not. And I just think that tolerance and withdrawal are normal, frequently seen phenomena, and we all should be aware of that.
I just have a quick addition that's not necessarily a specific area, but I think that your comment is on point, Dr. Keefe. I think that as we sat back and we had these experts looking at all of the data, one thing that was that was ubiquitous is there was this feeling that we could move away from this more traditional safety research and focus more on these sensitive subpopulations, and that was a key finding that we reported out. And we feel like we can now shift the caffeine research a bit.
Mm-hmm.
So the easiest low-hanging fruit in my domain of calcium and bone is to do acute studies related to calcium, urinary calcium excretion, and outcome measures that could be determined with fairly small intervention periods, including blood pressure, biomarkers of bone turnover, to get a little bit more functional. But the real gap everybody was mentioning was children and adolescents. So you could do these dose-response acute studies in adolescents to get at intermediate markers of exposure and functional physiological consequence. In terms of the massive cohort things with fracture and whatnot, I don't know if you're ever going to be able to assess intake all that well unless you take a duplicate. Standardized drinks, a can of a sugar-sweetened beverage with caffeine or energy drink with caffeine or something, that can be in the databases. But your own brews, there are a few studies where they actually are supposed to bring in a representative cup of their coffee or tea to get measured, and it'll take that kind of level of rigor to get exposure.
From an epidemiologic methods perspective, I think one thing that we can do when evaluating observational study designs is utilize quantitative bias analysis, which can take advantage of information from validation studies and apply that to results to produce adjusted measures that can inform. It doesn't remove the bias, but it informs us about the potential magnitude and direction of bias and gives us more insight into the data.
Okay, sir, please, if you'll identify yourself.
Yeah, thank you. Jeff Smith, the Ilse Southeast Asia region. Fantastic study, fantastic presentation of the methodology and the complexities involved. One quick comment, it's interesting to see the interplay between the toxicology and assessment of this type for safety of a particular compound. So caffeine is a little bit different than some because it can either occur naturally or be an additive. We don't have many tests generally for food additives that reach human toxicological levels. So how that fits in the whole toxicology issue is an important one. But the specific question, starting with Dr. Weaver, but maybe others have comments for their particular sections. In your presentation, you mentioned, I think, only 2 randomized control studies, and the rest is based on self-reported intake data. And I'm wondering if you saw close consistency between those two, because we know self-reported data is not that accurate sometimes, or at least not reproducible. So I'm wondering how close the correlation was between the randomized control trials, of which there are only 2, and the other broad levels of self-reported data.
So it's complicated because the 2 randomized controlled trials were in different outcome measures than all the others, and that was the calcium homeostasis compared to bone mineral density or fractures and falls. But in the end, we concluded that the comparator was safe, so to that extent, they were consistent.
In the back, please.
Hi, my name's Michael Gray. This is kind of a naive question, but is to infer anything about mechanism, have you guys ever considered, I mean, caffeine's a really dirty drug. It affects a lot of stuff, right? So have you ever considered for cardiovascular problems to target a subpopulation of people on calcium channel blockers to kind of infer anything about mechanism, or is that just not possible in this type of study?
Okay.
Does that make sense?
So if I understand correctly, you're saying, should we look more at mechanistic studies to get at thoughts earlier in the safety?
Well, but do an I can't even say it, this epidemiology study or whatever, but look at a subpopulation of like cardiac patients. The problem is there's a lot of confounds to that,
Right.
and just say, oh, well, normally, phosphodiesterase inhibition isn't going to be a mechanism in humans, but now they're selling like energy drinks, and I'm sure if you're a heart patient, you're probably not popping them. But I mean, is that there could be an interaction with the medications that people are on, is what I'm claiming.
Right. No, it's a very good comment. So our review looked specifically at exposures where there was not a co-exposure with another medication.
So you rooted that out.
Right.
But it'd be
However,
interesting.
Absolutely. It's something that's a potential future direction. It's something that Dr. Keefe also mentioned in the early opening, and something that the EFSA assessment also looked at. But as you can imagine, that it gets very complex because there's a lot of literature showing different
And you're destroying
different
your end.
exposure, right? And mechanistic events clearly play a key role.
Because I don't think like a lot of this stuff is going to be necessarily linear, because, I mean, the fact is there's so many uncontrolled variables and subpopulations within it. If I'm on a calcium channel blocker, you may not see any cardiovascular problem with me. I'm probably not going to be abusing energy drinks, but, there's a lot of confounds.
Right.
But thank you.
Right. I think you've done a good job summing up one of the future directions where we think that research could be focused now that we have a good baseline understanding of caffeine alone.
I'm assuming it's...
Please.
This is Jaspreet Ahuja from USDA. And first of all, thank you so much for a very comprehensive review and presentations. I actually have 3 questions, if that's okay. First is, I was just curious that while going through the reviews, were there any indications or results or studies that looked at the source of caffeine and its impact? Because I would assume caffeine obtained from coffee may have a different impact than from a 5-hour energy shot. So were there any of those studies?
I'm just asking, why would that matter? It's chemically distinct, does it?
I think that's your point is well taken. It's certainly caffeine in the blood is caffeine in the blood, but when you're not really looking at the effects of caffeine, you're looking at the effects of a cup of coffee or the effects of an energy shot. There are other substances in those beverages, and your point is well taken. That was not considered in this review.
Also, I'll build on that a little bit, that's something we were cognizant of. We did not do, for example, subgroup qualitative assessments based on source. We recognize the source, and I think probably the biggest differences in source that we saw were the sources associated with acute exposure, some of the acute outcomes versus some of the other outcomes. But that is also something that was evaluated in the risk of bias assessment, was essentially the purity, or, what are you looking at? Are you exposed to pure caffeine, or are you exposed to something like coffee or an energy drink that has other substances in it? And so that was accounted for when we were looking, but it's a great comment.
But you didn't see any study specifically looking at that, did you?
Right. We didn't look at that specifically.
Okay. My second question is that you mentioned that there might be some sensitive subpopulations. So was there a consideration, instead of giving one 400 milligram value, to maybe give a range?
That's a good comment, and it builds on some of the discussion that we've talked about here about personalized medicine and what's right for you and what might be right. And I think that's a good question, something that some of the regulatory agencies or people setting acceptable daily intakes may consider in the future. And I think the other big component, which Dr. Tenenbaum also discussed, was the idea of the temporal consideration, how much in a day versus how much in a sitting. And so some of those considerations may be included when there are some future recommendations or limits, safe values set.
If I can comment as well. What we're really addressing now is pharmacogenetics, and the problem with a range, it would not be universally applicable because of certain populations where the gene may be more or less prevalent. So your question is well taken, but I think it would be a challenge scientifically to address that, because in terms of generalizability, in one geographic region populated by various ethnicities would likely be different than in the second geographic area populated by different ethnicities. That's always the challenge when we're dealing with pharmacogenetics.
I guess that is the same consideration with one endpoint versus a range too, right?
I didn't understand or maybe I didn't hear your last comment.
I guess that is the same consideration with having a range versus having one single endpoint. I mean, I agree, there are so many factors that can impact that.
I think we can go on and on with this for quite some time, but I don't think in trying to establish some sort of levels of concern, whether they be for cardiovascular, behavior, acute toxicity, or what have you, is best served with coming up with a range that has limited generalizability. We have to recognize that there's going to be people that are sensitive and would fall out of that, and there would likely be people that are resistant as well. You're striving for perfection, and that's marvelous. I don't know how far we can get to that goal.
True. And my last question, when you were looking at coffee, like coffee, tea and all, and I saw supplements, there are also a lot of, I see in supplements things like guarana added, which are very high in caffeine. Was that taken into consideration in determining the amounts?
To determining the amounts of caffeine?
Yes, consumed.
So in our dosimetric decision tree, essentially, we categorize what we would count as a source and not. And so we tried to spell all of that out, very specifically what we would accept and not accept and how it may impact. So I honestly just can't remember off the top of my head exactly how that was handled, but it's spelled out in the supplemental materials.
Thank you very much.
It's in the supplemental materials of the manuscript, supplemental line.
Thank you. Dr. Main?
Hi, I'm Susan Main. Thank you. I look forward to looking at the report soon. So I was intrigued by the blood pressure data and all the variability there, and you made the comment that that exercise raises blood pressure, caffeine may be elevating blood pressure. Were any of the studies done in exercising individuals? And do we have any ability to look at kind of the joint effects of caffeine and physical activity together? Because one of the areas we know it's a growing segment of the caffeine marketplace is, used for physical performance, weight trainers, athletes. So is there any data on that, or is that a research need to look at joint effects of caffeine and physical activity?
Well, I can answer broadly. Unfortunately, our experts aren't here, and I know they know this off the top of their head, but much of our controlled trial data was in exercising individuals or athletes, military, et cetera. So I think the data do exist to do that. We did not investigate that combination per se, but I think that data is there.
Where it might be more interesting, I'm taking this from a recent paper I'm going to tell you about, is response to the interaction and the response to diet, rather than you're talking about cross-sectional relationships. But in the GenSalt, that large study in China, that's primary purpose is to look at genetic variation in response to sodium reduction. One of their spin-off papers looked at dividing up the population in terms of like quartiles, I think, of physical activity, and the ones that were more physically active had a much greater response in blood pressure reduction to sodium reduction. It's an amazing study because they took 1,800 people and had them on a high salt diet for 1 week, and then they changed them to a low salt diet. So they're looking at the response, and physical activity really influenced how great that response to diet. And I think that's kind of maybe the way to look at what you're suggesting. If you have a various level of physical activity, what's your response to caffeine in terms of blood pressure?
Thank you. Well, if there are no further questions, I'd like to, again, thank all the panelists for the very wonderful presentation this morning, and we look forward to looking at the report. Thank you.